Welcome. I'm Dr. Steve Hanauer, Professor of Medicine and Director of the Digestive Health Center at Northwestern University, and I'm joined by Dr. David Rubin, Professor of Medicine and Chief of the Section of Gastroenterology, Hepatology, and Nutrition at the University of Chicago. Today's discussion is going to focus on considerations in the use of biosimilars for gastrointestinal disorders, but we are going to focus on inflammatory bowel disease, where the two currently approved biosimilars are actually going to be utilized. Our disclosures can be seen on the next slide, and I would emphasize that this program is supported by independent educational grants from Boehringer Ingelheim Pharmaceuticals, Inc. and Pfizer, Inc. At the conclusion of this presentation, we expect that the learners will appreciate the landscape that has contributed to the emergence of biosimilars in the U.S. inflammatory bowel disease market. We hope to gain clarity on the definition of biosimilars and the abbreviated FDA approval pathway, appreciate the rationale for extrapolation of indications. We want to recognize that no biosimilar has received an interchangeable designation, and we will discuss the definitions of interchangeability. And we want to consider the clinical scenarios in which biosimilars may be utilized safely. To begin with, some economic considerations of biosimilars. And indeed, the whole rationale for biosimilars comes from the Affordable Care Act, which recognized the significant cost of biologic agents in the United States. And as this slide demonstrates, while only 1% of prescriptions are for biologic agents, biologics count for 28% of our current drug spending. Indeed, biologics are very expensive to develop and to use. It's been estimated that the cost for a biologic agent in the United States is approximately $45 per day, which is compared with only $2 a day for other chemical agents. The global sales of biologics are expected to reach $180 billion within this calendar year, and to date, 11 biologic agents account for about 50% of the entire cost. These agents continue to lose exclusivity, and we will be discussing one agent, infliximab, which has already lost exclusivity, and another agent, adalimumab, which has been approved by the United States FDA, but has not yet lost exclusivity according to patents. It's been estimated that the savings in the United States and European Union with biosimilars is going to be between $56 and $100 billion over the next 5 years. As I mentioned, biologics are expensive, both biologics and biosimilars, and cost an estimated $1.2 billion for the originators to develop. Now, the exact cost is really undisclosed because insurance companies receive both discounts and rebates that are confidential. But it's been estimated that approximately a 30% potential savings will be incurred by switching to biosimilar agents. And to date, price reductions have varied considerably between the European countries where biosimilars have made inroads, with up to 69% cost savings seen for the biosimilar to infliximab in Norway. These costs and potential cost savings have led to significant development for biosimilars with both adalimumab and infliximab within the United States. And as I mentioned, we'll be discussing two biosimilars that have been approved for infliximab, and while one for adalimumab has been approved, it is not yet marketed in the United States. But several other industry companies are developing biosimilars within Europe, Asia, and within the United States. And Dr. David Rubin is now going to be discussing some of the definitions used regarding biosimilars.
Thanks, Steve, and thanks everyone for joining us for this discussion. Biologics are different from small molecule therapies in that they are proteins. And as demonstrated on this slide, you can appreciate that the monoclonal antibodies are huge structures compared to small molecules, the example given here being aspirin. And the distinction here is that a small molecule, because it's an organic chemical, has a well-defined structure, can be delivered orally, has a well-described metabolism and metabolic pathway, and because it is a chemical, can actually be produced in an identical fashion in what we call generics. In contrast, biologics, because these are proteins, have a variable 3D structure, must be delivered parenteral in part because of their protein nature, but also because they're so large they can't be absorbed through the small bowel. They have degradation by catabolism, and they're obviously very expensive, but when you try to reproduce them, you end up with what are known as biosimilars rather than identical copies. The FDA defines a biosimilar as a biological product that is highly similar to a reference product, notwithstanding minor differences in clinically inactive components. If you recall from your days in biochemistry class, protein is constructed from its primary structure of amino acids through to its quaternary structure of protein folding and post-translational glycosylation. Therefore, there are some minor differences that occur when a biosimilar is created. However, and very importantly from the FDA point of view and other regulatory agencies around the world, there must be no clinically meaningful differences in terms of safety, purity, and potency. So the similarity is in how the agent works, but isn't identical to the originator. Sometimes the originator is also called the reference product, so you may hear those used interchangeably. And in fact, as we discuss the originator and reference products that are currently available on the market for inflammatory bowel disease, you can also appreciate the discussion that's occurred that some of the originators and reference products have in fact been biosimilars to themselves as they went through their production over many different years. There are a number of practical considerations for biosimilars. The first is to realize and understand that the dosing is the same as the reference drug because it obtains the same labeling as the reference drug. It is also expected and will be the case that pre-authorization and reauthorization will occur for these agents, just like we currently do. It's not necessarily going to be the case that the way we get our patients to receive these therapies will be simplified or changed in any way. It is also a common question, and it's important to know, that the existing commercial therapeutic monitoring assays to measure drug levels and anti-drug antibodies are expected to work with the biosimilars and can be used the same way. And it's also important to recognize, and this is something new for us to consider since we haven't had this in our field before, that the anti-drug antibodies between a biosimilar and the reference or originator will cross-react. In other words, if you have a patient on an originator drug and they develop anti-drug antibodies that neutralize or lead to rapid clearance of that drug, giving the patient the biosimilar will have the same reaction. And in the past, when we've had a patient lose response or develop anti-drug antibodies to one drug within a class, when we cycle to another drug within the class, we didn't have to worry about cross-reactivity. But it should make sense to you now that I've defined this, that the biosimilar will react the same way and anti-drug antibodies will cross-react. Steve will now cover the regulatory pathway of biosimilars in the U.S.
So in contrast to the development of an originator drug, which relies on both preclinical and analytical data as well as PK and PD, the major cost and the major aspect of the development of the originator pertains to clinical studies which must be performed in each and every indication for which the company or the originator plans to market the drug. In contrast, biosimilar development pathways are different. They rely on the increased amount of preclinical and analytical data that will demonstrate biosimilarity, as well as PK and PD data that also establish biosimilarity. In the clinical studies performed for a biosimilar, it is only necessary to select one or two indications that will be representative of the grouping of indications for the originator to study, and each indication is not necessary, as we will discuss, related to extrapolation. Along these lines, the FDA, the European medical authorities, and Health Canada will evaluate what's known as the totality of evidence to demonstrate biosimilarity. These include a stepwise approach by our FDA, in vitro structural analyses that look at glycosylation and other functional assays, in vivo studies that also include additional functional assays according to the different mechanisms of action, toxicity studies, pharmacokinetic and pharmacodynamic attributes, as well as immunogenicity. As mentioned, the clinical studies may be more limited to a single indication or additional indications that will include the pharmacology data as well as comparative clinical studies related to immunogenicity. Now let's discuss several different types of studies. Traditionally, originator drugs use superiority studies, for instance, the drug versus placebo, to demonstrate superior efficacy. Non-inferiority studies are performed when a proposed product is not inferior to an unacceptable extent. For biosimilarity, primarily equivalence studies that are intended to demonstrate both the proposed product is not inferior and is not superior. If it is either inferior to the originator or superior to the originator in clinical studies, it would not be considered a biosimilar and would actually be a either non-acceptable product or a new drug. If biosimilars had to perform clinical studies in each and every indication, there would be no cost savings for the development of biosimilars. And indeed, the concept of biosimilars are to reduce the cost and improve access for patients. This requires the concept of extrapolation, which means that clinical trials in one indication can be used as a rationale for clinical use in other indications for which the originator biologic product is approved. And this does require rigorous, appropriate scientific justification. The FDA has determined that extrapolation may be possible if the overall similarity with the originator product is demonstrated based on the totality of evidence, that clinical similarity is demonstrated in a key indication, and that the mechanism of action and pathophysiology of the other indications is supported by the data. Our regulatory agencies have addressed key concerns regarding extrapolation, that it is considered on a case-by-case basis, particularly where the mechanism of action for a condition is not understood. Separate trials may be necessary. They've stated that superimposable biologic data must be provided that address every functional aspect of the agent in question. So, for instance, with a TNF agent, is the function both binding or neutralizing TNF in the serum as well as TNF that's exposed on surface cells. In addition, data should be generated in patient populations and utilize a clinical endpoint that is sensitive enough to detect clinically meaningful differences in the indication. And consideration must be given to comorbidities and concomitant medications as well as inter-subject variability. There are several different studies types that have been performed. These include transition studies where patients are randomized to drug or biosimilar and then transitioned to the biosimilar. Substitution studies where patients are initially randomized to drug or biosimilar or originator and then switched in a single time, or multiple switches may be used in what would be considered an interchangeability study where patients are exposed to the originator, then the biosimilar, back to the originator, then the biosimilar, which might force issues such as immunogenicity to become apparent. In addition, the FDA guidance for biosimilarity also requires marketing for post-marketing monitoring. And this may be required for any biosimilar. It will be based on the safety concerns both associated with the originator and the broader class of the biosimilars. And adequate mechanisms are required to distinguish adverse events associated with the proposed biosimilar versus the originator. And we'll talk more about labeling in a minute. Now, biosimilarity in and of itself does not determine interchangeability. Interchangeability has been defined by the FDA as a designation that allows for back and forth switching between biologic agents without loss of efficacy or increased risk for adverse events and without the input from the prescriber. It is a separate designation beyond approval and extrapolation. And I should point out that to date, no biosimilars in the U.S. in any disease state has received interchangeability designation. In order to show interchangeability, industry must, among other things, show that the immunogenicity risk of the product in different patient populations is not different. So the agency, the FDA, has come up with biosimilar nomenclatures that are intended to avoid inaccurate perception of biosimilar efficacy and influence prescribing practice of biosimilars. So each biosimilar is to receive a 4-digit extension. Infliximab as Remicade is determined as HJMT. Most recently approved Inflectra and Renflixis have the 4-digit designations DYYB for Inflectra and ABDA for Renflixis to allow monitoring of the specific agent that has been administered to any individual patient. David, why don't you discuss what's the status of biosimilars in the United States?
Thanks, Steve. So to date, as of the recording of our call at the end of May 2017, the status of U.S. biosimilars in IBD is as follows. There are 3 biosimilars that have been approved by the FDA with extrapolation from studies in other disease states to be available for similar labeling of Crohn's and ulcerative colitis. As Steve mentioned, the biosimilars for Infliximab originator, which we know by brand Remicade, include Inflectra, which is now designated as Infliximab DYYB, This is approved by the FDA in April 2016 and has been available technically since November 2016 with an original projection of a 15% discount over the wholesale cost of the originator therapy. And it is now being used in some markets and by some infusion centers and health systems. And more recently approved in April 2017 is another biosimilar to infliximab called Renflexis, or infliximab ABDA. We aren't aware that this has yet been used or available in the U.S. market, but is expected to be so. The biosimilar for the originator to adalimumab, known as brand Humira, was approved in September 2016. It's known as Amjevita or adalimumab ATTO. This one is in legal discussions related to patents and some other issues relating to exclusivity, as Steve alluded to earlier. There have been a number of studies of biosimilar infliximab, the CTP13, which we know as the Inflectra or the infliximab DYYB, because the agent has been available in Europe and Asia for 2 to 3 years already. So our colleagues in those parts of the world have looked at the outcomes of their patients who've been exposed to these agents in treating Crohn's disease and ulcerative colitis. I'll start by mentioning this meta-analysis that was published in the Annals of Internal Medicine last year, which looked at all biosimilars for all indications, not just GI diseases, but including the other reasons to treat with anti-TNF biologics. And among the 19 studies that compared biosimilars and their referenced biologics, there were 8 phase 1 trials, 5 phase 3 trials. Recall that you do not need to perform phase 2 trials with biosimilars, and 6 observational studies included in this meta-analysis. The margin of equivalence was between 80 and 125%. There were no new safety signals as would be expected with biosimilarity, and the conclusion from the study was that preliminary evidence supports the biosimilarity of biosimilars. The authors also chose to say that they thought the preliminary evidence supported interchangeability, but I want to point out to you that there had been no study to date, and there has been no study up till now that shows the interchangeability as defined by the FDA, as Steve had described to you. More recently is a meta-analysis of CTP13, infliximab biosimilar, in IBD alone. Of 11 published studies and abstracts of 829 patients, the efficacy and safety of the CTP13 in Crohn's disease and ulcerative colitis were similar to historical registration studies. It should be noted that this has included in some of these studies an assessment of anti-drug antibody development, which also was similar to prior studies that have been published. So far, based on the available data in Asia and Europe and these 2 meta-analyses, the biosimilar to infliximab is functioning as expected to the originator in Crohn's and UC. Steve, did you want to describe the NOR switch study?
Certainly. The Norwegian government several years ago required that all patients being treated with infliximab be put into a clinical trial over 1 year to look at the potential for switching between infliximab and the biosimilar CTP13. The primary outcome was disease worsening at 12 months, and anti-drug antibodies were also assessed. Now, there were multiple indications for infliximab in this NOR-switch study that included other rheumatologic and dermatologic indications, including rheumatoid arthritis, psoriasis, and other indications. But the study was important because it did include approximately 250 patients with inflammatory bowel disease. As you can see, 155 with Crohn's and 93 with ulcerative colitis. When the patients were switched, according to the primary outcome criteria for the study. There were not significant differences between Remicade and CTP13 according to clinical results or to anti-drug antibodies. It is admitted that the Crohn's disease criteria reached the borders of the criteria for a primary response, but was considered to be appropriate for the clinical trial. And David's going to discuss some of the data presented at ECHO.
So at the European Crohn's and Colitis Organization meeting in 2017, there were many abstracts looking at ongoing experience with the biosimilar to infliximab in the different parts of the world. I want to point out that to date, there's really only been single switch, or what are sometimes called transition studies, where patients who are stable on the originator infliximab are transitioned to the biosimilar infliximab, or studies in which patients receive the biosimilar infliximab as new starts. And the ongoing experience has continued to demonstrate what we would expect with these agents. In this study, from 58 different centers in 16 countries of 220 patients who were randomized, you can see that there was a similar and consistent trend in proportion to achieve a Crohn's disease activity index response and clinical remission with both CTP13 and the originator infliximab treatments. This continues to demonstrate the relative similarity in real-world experience with these agents, or with this agent. In the second study we've chosen to share, called the PROSIT study, is a cohort of patients who received the infliximab biosimilar, and it was a prolonged follow-up from Italy of 680 consecutive patients with Crohn's disease and ulcerative colitis from both academic and community-based centers. 400 of the patients were new starts to anti-TNF, and the other patients had been previously exposed to one or more anti-TNF biological agents. 109 of the patients were switched after a mean of 18 plus or minus 10 previous infusions of infliximab, and over 4,000 infusions were recorded. This is of interest as one of the largest prospective cohorts of patients who were treated with CTP13 both as a new start as well as a transition therapy, and there were no further signals of difference in safety and clinical efficacy in either the Crohn's or UC patients. Further support for these agents' safety. And lastly is just what's happened in these markets over time. We've come to appreciate that the physicians, and specifically gastroenterologists, who were not confident in the use of the biosimilars or were concerned about potential risks of these agents, have actually, after using them, developed much more confidence and comfort in their use. It's also been seen that the pattern of use in this specific audit, which was from the United Kingdom, has demonstrated that because the agents were less expensive and there were more agents available, they've actually started to result in earlier use of biological anti-TNF therapy in milder disease. So one of the interesting points that has come up is if people become more comfortable using these agents overall, and when we have more agents available, will any potential cost savings to society or to health systems be neutralized by increased use of the drugs altogether? You can look at sales and spending with the uptake of biosimilars in 23 countries within the European agency, and you can appreciate the growth in spending of biosimilar dollars along with a slight decrease of the reference product and the slight increase overall of the total spending. So in other words, as people are transitioning to biosimilars from the use of reference products which were priced higher, the overall volume has increased, and therefore the overall cost has actually not gone down. It's actually increased somewhat. Now, if you are most interested in how patients are doing overall, this is probably representative of more patients getting effective therapy and hopefully doing well. What isn't adjusted here is also the appreciation that there's been a rise in incidence of of IBD that may go along with these measures over time as well. So the question becomes, who would actually benefit from a 20 to 30% cost savings, specifically in the U.S.? We actually have some cynicism about this, that this would be some cost shifting, and that any savings may be individually by insurance companies or by infusion centers. But it's not clear, and there's no evidence to suggest that patients will have a lower copay or that our access to prescribing them will be any easier or different. So we'll have to see how this changes over time, but I would say stay tuned on whether this is actually going to save money in our markets in the United States. Steve, do you want to discuss some of the clinical scenarios where biosimilars might be used and give some practical points?
Absolutely, and I think it's time to get down to earth as we are all being exposed to the increasing availability of biosimilars in our different regions and in our different health systems. And of course, this is going to be dependent on the health system and the third-party payers who are determining the use within the individual system formularies. But let's consider several different scenarios. First and foremost, a new start, a new patient who one is deemed should get a biologic agent for inflammatory bowel disease or another indication. The first thing that we need to recognize is that there are some differences between Europe and the United States. In Europe, the drugs are regulated by country, and each institution has a limited amount that they can spend, but the determination of the prescription is by the clinician. In the United States and in our current system, while we may desire to initiate a biologic agent for an individual patient, it is most customary that our third-party payers are looking to the labeling of the originator to allow approval of the use of the agent. So in other words, we may wish to start a patient earlier, but unless they have moderate to severe disease that has not responded to conventional agents, we may not be able to prescribe off of the labeled indication. This is our current status and what we're dealing with the originators. So the concept of having a less expensive biosimilar is not likely to allow us to use it off of the labeled indication. The prescriber will have the choice of reference product or biosimilar at the present time, but the cost to the patient may be determined by the third-party payer. So new starts, I think very few of us have concerns whether it is the originator or a biosimilar, because patients should be then getting consistent drug that has been shown to be biosimilar. Now, David mentioned primary non-responders or immunogenicity. If a patient does not respond to the originator, there's no rationale why they should respond to a biosimilar. It would be necessary to switch mechanisms of action, as we have learned within the situation of primary non-responders to biologic agents. Now, what about a patient who's stable, where the third-party payer would like to suggest a non-medical switch? In other words, the patient is stable on the originator, but the payer would like to switch to the biosimilar. At the present time, it is required that the physician signs off on this switch. It is not interchangeable, as we've repeatedly said, and interchangeability would allow a pharmacist to switch without the providers, without the clinician's approval. That is not the case now. The non-medical switch would be determined on cost only, and depending on the individual system or the individual insurance situation, the cost to the individual patient may stay the same or increase or decrease depending on the selected agent. Then finally, if a patient loses response and the loss of response is due to anti-drug antibodies or immunogenicity, as David said earlier, switching to a biosimilar will not offer any advantages. On the other hand, if it's due to low drug levels, either raising the dose of the originator or a higher dose of the biosimilar would be appropriate. But if there are adequate drug levels and the patient loses response, switching to the biosimilar will again not have any advantage. So hopefully those are some down-to-earth examples of where we may see biosimilars applied within our clinical usage. And David is going to now conclude our program with final statements.
Thanks, Steve. So today we've reviewed biosimilars in GI, but really focused on inflammatory bowel disease because to date that's where they are approved in the United States and actually through Europe and Asia. Biosimilars are not generics. We should avoid using that term, and hopefully you now understand that these are complex protein structures that are similar in their clinical efficacy, safety, and purity. The FDA allows for extrapolation of indication for biosimilars, which means that they can be studied in other disease states and then gain approval in Crohn's disease and ulcerative colitis. And the available data in Europe and Asia that we've shared summaries of with you today suggest that biosimilars to anti-TNFs behave similarly to their reference products. So far the data is all for the infliximab biosimilar because that agent has been available first and for several years. The drug assays for the reference products are expected to work similarly for the biosimilars, so you should be able to obtain drug levels and anti-drug antibodies using the same assays. Remember that anti-drug antibodies to the reference products or to the biosimilars will cross-react to the biosimilars or to the reference products. This is an important consideration because we don't expect payers to necessarily understand this. So if you want to switch your patient to another anti-TNF because they've developed what you believe to be or have proven to be immunogenicity, you need to be very specific and not allow them to just switch to the biosimilar because the patient will have the same reaction or worse. No biosimilar in the U.S. has yet obtained an interchangeable designation. The available European and Asian data suggests that it's at least safe to do so in a single transition, but there is not yet a study that demonstrates back and forth switching to make us most comfortable. And as we see more biosimilars coming to the market where there really will be multiple agents and ongoing price wars, it will be very, very important for us to understand this further. We hope that you have enjoyed this presentation and that we've added to your knowledge and comfort level regarding these agents. We anticipate that there will be additional questions as the drugs are being used throughout the United States, but we'll do our best to keep you updated and we'll look forward to future programs. On behalf of the AGA and on the Crohn's and Colitis Foundation, thank you very much.